In this video, we will discuss about standard CAR-T cells and stem cell memory T cell based CAR-T cells characterization. So, in the panel A, it shows how these different kind of CAR-T cells are formed. For this standard CAR-T cells, they used a mixed population of these T cells and then transduced them with this retrovirus, this targeting CD19. Along with that, they used anti-CD3 and IL-2 to activate these T cells populations, and these standard CAR-T cells can then be used in the 20 different patients. Whereas in the stem cell memory T cell based CAR-T cells, so what they did is they, after leukapheresis, they first enrich the CD8 plus naïve CD8 plus T cells, and this is based on the previously published protocol. So this from these naïve CD8 plus T cells, they also did the retrovirus transductions targeting CD19. Along with that, for this activations of the naïve CD8 plus T cells towards the stem cell memory T cells, they used anti-CD3 and anti-CD28 beads. Along with that, to prevent the undesired differentiation, they also used IL-7, IL-21 and TWS119. So from that they get a population which mainly contains the stem cell memory T cells, and they use this population, this CAR-T cells, on 11 different patients. Results for how they react to the patient is not shown in this figure. Here it shows the characterizations. So after these two populations are formed, they first checked the CD8 plus and CD4 plus populations. As expected in the standard, they see both the populations for the CD8 plus on the y-axis and CD4 plus on the x-axis. Whereas for the case of these stem cell memory T cells, they only see CD8 plus T cells because their populations for the transduction is CD8 plus, so they mainly see CD8 plus T cells. After that, they further characterized to check this CD45 and this CD45R0 and CD45RA. For the case of CD4, the standard CAR-T cells, they are able to see the mixed populations, whereas in the case of T cells, stem cell memory T cells, they are able to see the specific populations. After that, they quantified this CD8 plus T cells and CD4 plus T cells in two different CAR-T cells, and they are able to see that for the case of standard CAR-T cells, almost same amount of CD8 and CD4 plus T cells are formed, whereas for the case of this CAR-T cells that are formed on stem cell memory T cells, they are able to see a significant enrichment of CD8 plus T cells and almost negligible amount of CD4 plus T cells, indicating that this stem cell memory T cells is more homogeneous. So afterwards, they checked the other populations also in the panel D, and from that it is clear that the Tscm based CAR-T cells are enriched in this Tscm, whereas other populations are low. Whereas for the case of standard CAR-T cells, it is divided into different populations, with this effector memory T cells and terminal effector T cells contributions are slightly higher than the other populations. Thereafter, they did the phenograph analysis on both the CAR-T cells to find out the different populations, and they are able to see 10 different clusters. And out of these 10 different clusters, they are able to find out that this panel 4, 6, 8, 9, 4, 6, 8, 9, and these populations belongs to the CAR-Ts that are made from Tscm, and these things they are further quantified in the panel F, that these four populations contribute higher. 4, 6, 8, 10 contributes majorly in this stem cells, CAR-T cells derived from Tscm, whereas standard cells has the different proportions of most of the populations. Afterwards, they also did the heat map to show heat map, and here the results also shows that the clusters 4, 6, 8, 7, 4, 6, 8, 9 are less differentiated, or they are less differentiated among each other, whereas the other populations are differentiated comparatively higher, indicating that the CAR-T cells that are made from stem cell memory T cells are more homogeneous compared to the case where standard CAR-T cells compared to the process compared to the standard CAR-T cells, indicating that the results from this Tscm based CAR-T cells could be more reproducible. This is what and this last sentence is from my side. So this is what the figure shows, how they this is what the first figure of this paper shows, that shows the characterizations of this standard CAR-T cells and stem cell memory T cells based CAR-T cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.